Hello everyone. It's my great pleasure to share our newly published work, in which we uncover a remarkable biological system in nature: how deer antlers are able to elongate bony tissue by centimeters each day. Deer antlers elongate 2 centimeters per day, far exceeding the 2 centimeters annual growth of the human growth plate, making them the fastest bone regenerating organ in mammals. Their growth mode challenges the classical view of endochondral ossification. This remarkable process is driven by the antler growth, which contains abundant mesenchymal-derived stem-progenitor cells and a highly vascularized, metabolically active cartilaginage. Together, these features create a continuous and rapid ossification system that supports intense proliferation while avoiding malignant transformation. However, the many cell types within this niche coordinate to sustain such extreme elongation has remained largely unknown. We sampled 5 tissue layers from the distal to proximal regions of the growth center to establish a spatial framework. High-quality multi-omics analysis identified more than 10 major cell types, including stem cells, progenitor cells, chondrocyte at multiple stages, osteogenic cells, and these datasets show strong consistency, and spatial mapping revealed a clear differentiation trajectory from stem cells to cartilage and then to bone. Among these cell populations, progenitors contain a highly proliferative subcluster that drives the centimeter-scale daily elongation of antlers. Stem cells secrete IGF-1, PTHLH, and other signals that boost proliferation while blocking premature differentiation, thereby keeping progenitor pool active yet tightly regulated. Spatial and functional analysis further show that coordinated stem-progenitor crosstalk is essential for rapid growth. Despite their rapid cycling, these cells are transcriptionally distinct from osteosarcoma, and antlers ensure growth safety through a balanced proliferation apoptosis program. We observed a typical hypoxic niche near the proximal zone along the differentiation trajectory, whereas the distal region is enriched for pathways related to vessel branching and maturation, forming a stratified, dynamically renewing vascular network. This differs sharply from the completely avascular growth plate. Vascularized cartilage confers 3 major advantages: improved metabolic support for chondrocyte, rapid additive cartilage expansion, and facilitated delivery of osteoblasts to mineralization fronts, allowing highly efficient bone formation. We identified a PHEX-positive chondrocyte subpopulation positioned near hypertrophic cartilage. These cells express bone mineralization genes and receive H-hat signals from nearby cartilage, suggesting a central role in rapid ossification. Their expression of MMP9 and MMP indicates functions in matrix remodeling, vascular invasion, and osteoblast recruitment. Transcriptional signatures show strong associations with osteoblast differentiation, pointing to an osteoblast-like intermediate state. Because RUNX2 is highly expressed in antler progenitor and hypertrophic chondrocyte, and given that hypertrophic chondrocyte in mammals can transdifferentiate into osteoblasts, we propose a PHEX-positive intermediate in antler chondrocyte-to-osteoblast conversion. Using antlers spatial organization, we identified 2 osteoblast types: OBD adjacent to PHEX-positive cells and OBP not adjacent. Pseudotime analysis revealed a trajectory from chondrocyte and hypertrophic chondrocyte through PHEX-positive cells toward OBD, with increasing osteogenic markers and loss of hypertrophic signatures. Xenographed nude mice further confirmed the osteogenic capacity of hypertrophic chondrocyte. Taken together, the antler growth center employs a rare natural mechanism to achieve rapid, sustained, and low-risk bone regeneration. This system suggests that future strategies for tissue regeneration and bone repair may not need to rely solely on the classical growth plate structure. Instead, the antler model may inspire the development of more efficient and safer approaches for bone reconstruction.